This is no trick of the light. These rabbits' eyes and skin really do glow green. It's amazing what he's done.
This is transgenics in action. Scientists have managed to isolate a tiny bit of jellyfish DNA that makes them fluoresce, known as the GFP gene. Dr. Houdebine has simply taken that gene and inserted it into bacteria. As the bacteria reproduce, the gene is multiplied. And then these genes are injected into the fertilized egg of a rabbit. Dr. Houdebine then places the egg into the mother. As the egg grows to form the rabbit embryo, it copies the fluorescing gene into every cell. 31 days later, you've got fluorescent bunnies.
You may be surprised to hear that Dr. Houdebine does all this with jellyfish genes he orders off the internet.
Traditionally, a salmon only grows in warm water. But as soon as the water gets cold in winter, the gene controlling the salmon's growth switches off, and the fish stops growing. Not anymore. Joe takes the genetic switch which controls growth from another species of fish that grows in cold water. He inserts that into the salmon egg. The presence of this genetic switch means the new salmon now grows all year round, whatever the water temperature.
This animal looks normal, but it's anything but.
These pigs are unlike any other pigs in the world. They're different because they carry a very special gene that allows them to digest all of the phosphorus in their diet. The gene is constructed in the laboratory that we use.
The remarkable thing about the EnviroPigs is that they carry a man-made gene, which John has created from scratch.
One part comes from E. coli bacteria, which produces an enzyme that can break down phosphorus. And the other comes from a mouse, which produces a genetic switch that allows this special enzyme to be made in saliva. These two bits of DNA were combined to form a new compound gene, which was then inserted into a fertilized pig egg. Now every time John's pigs eat and produce saliva, they also make the phosphorus-busting enzyme, helping to make their poo more environmentally friendly. So just how do you create a clone? Expert cloner Earl Huang is keen to show Olivia just how it's done.
It's still kind of hard to see. Let me see if I can get it. Today we're working with bovine.
So it's a cow egg?
Yes. These eggs come from a slaughterhouse, and what I'm basically doing is taking out all the DNA and creating a blank for the actual cloning process to begin.
So in order to clone, you need an empty egg.
Exactly.
And so you're plunging this glass pipette into the egg and just suctioning out the DNA.
Yes.
It's an extremely delicate operation.
It is. You have to have good hand-eye coordination and have to be gentle with the eggs. You don't want to kill them.
And that's what's amazing about cloning. It doesn't require particularly complicated chemistry, just a microscope, a very sharp glass needle, and incredibly steady hands.
And I'll go in with the pipette.
And draw it up like juices from a turkey. I have to say, it's fascinating to see it.
Next, Earl has to hoover up the cells containing the DNA of the animal he wants to clone.
These are cells grown from the skin of the animal that we want to clone.
Exactly.
It amazes me that you can put a cell from a grown-up animal into an empty egg and something happens.
Well, the cell basically contains the whole genetic makeup of that animal.
You now insert the DNA of the animal you want to clone into the empty egg. One cell into each
You
egg.
can see there's a little pocket of space.
What I'll do is puncture the egg and just carefully deposit the cell in there.
Last of all, you need to wake up the egg containing the new DNA. All it takes is a spark of electricity.
Earl may make it look easy, but in fact it's immensely difficult to pull off in practice. Dolly the sheep was the only survivor of 300 attempts. So the scientists set to work to see if they could manage to make rice high in vitamin A by persuading it to produce the carotenoids. Their science was pioneering. They identified not 1 or 2, but 3 genes. 2 from the daffodil and 1 from a bacteria, which they thought would help produce carotenoids. They then tried to insert all 3 of these into rice DNA. In 1999, after years of trial and error, they finally managed to create rice that produced beta-carotene. They could tell that this was being produced because the genetically engineered rice was colored yellow. They christened it golden rice.
That was amazing science. It still is amazing science. At the time, no other genetically modified plant had more than 1 gene expressing. They put 3 in and turned on a pathway.